DNA. Algorithm affecting DNA structure, protein synthesis, and mutations. Structure. DNA stands for deoxyribonucleic acid. It is made up of building blocks called nucleotides, deoxyribose phosphate group, and one of each nitrogenous base, A, T, G, and C. A is adenine, T is thymine, G is guanine, and C is cytosine. A always corresponds with T, vice versa, and G always corresponds with C, vice versa. This correspondence is a hydrogen bond between two nitrogenous bases. At the molecular level, a DNA strand looks like a twisted ladder, or a double helix. Outer strands of the DNA are made up of phosphate groups and deoxyribose. Inner strands are the hydrogen bonds between the nitrogenous bases. There are bonds that occur between the outer strand. These are covalent bonds. Main property that helps the structure hold together are the hydrogen bonds, the nitrogenous bond bases, and the covalent bonds between the phosphate groups and sugars, deoxyribose. And finally, uses proteins to execute these biological activities. Protein synthesis. There are three different roles of RNA and its function in protein synthesis. These forms are mRNA, which is messenger RNA, tRNA, transfer RNA, and RNA, rRNA, which is ribosomal RNA. mRNA carries genetic information copied from the DNA in a series of three base code words, which specifies each amino acid. tRNA, transfer RNA, is what deciphers the code word in mRNA. Each type of amino acid has its own type of RNA, own type of tRNA, which combines it and carries it to the growing end of polypeptide chain if the next code word on mRNA calls for it. The correct tRNA with its attached amino acid is selected at each step because each specific tRNA molecule contains a three-base sequence that can base pair with its complementary code word in the RNA. Ribosomal rRNA, associates with a set of proteins to form ribosomes. These complex structures, which physically move along an RNA structure, charges the assembly of amino acids into protein chains. They also bind tRNAs and various molecules necessary for protein synthesis. Ribosomes are composed of a large and small subunit, each of which contains its own RNA molecule or molecule. Mutations. There are many types of mutations, such as 1, the replacement of one base group for another, 2, the removal of one or more base groups, and 3, the injection of one or more base groups. There are two types of replacements that are possible. A transition is the substitution of one purine by the other, without one pyrimidine by the other. In contrast, a transversion is the replacement of a purine by a pyrimidine, or that of a pyrimidine by a purine. Our scientific argument is that the cause of mutation is by the substitution of one base pair for another, the deletion of one or more base pairs, and the insertion of one or more base pairs. The potential impact of the mutation is that if the cytoplasm receives the new mutation, the cells will never stop working and will result in a very long amino acid change.